All right, so thanks for squeezing me in here. So I just want to say population cycles, ubiquitous in macro systems ecology, but also in microbial systems. So we've got the E. coli and T4 bacteriophage here. And we have a lot of theoretical explanations for population cycles. Most trivially, periodic forcing. Most famously, I would say predator-prey dynamics, and then a bunch more obscure mechanisms. But notably missing from this list, mutualism and cross-feeding in particular. And so we want to see if mutualistic interactions can lead to population cycles. And I'd done a lot of work before on cross-feeding. We found lots of interesting things, alternate stable states, spatial patterns, self-organization, so on. But one thing we did not find were population cycles. And so I didn't think it was going to happen, but then I got an email from Ophelia Venturelli, who had set up an experiment with her master's students, Tyler Ross, where they'd engineered a cross-feeding system of E. coli, which cross-feed two amino acids and compete for glucose. And it more or less matched our previous theory. Put in a serial batch system in the lab, and let's see what happens. So for a particular supply point or supply ratio of nutrients, it goes to a stable equilibrium. If you don't supply externally any amino acids, again, goes to equilibrium. But in between, what's this? Period 2 cycle. Where did this come from? So that's what we wanted to know, particularly because our previous theory that more or less seems to match would not produce such dynamics. And it's not just a fluke, it was robust across a range of environmental parameters and replicable. So we figured out that the missing ingredient here is how the microbes excrete their amino acids. So in my previous models, they were on autopilot, they would just continuously excrete. What we put in our new model is what we call limitation-dependent excretion. So if this particular microbe is limited by glucose, they take up the glucose and amino acid that they require, and they synthesize amino acid proportional to their glucose intake, and that's it. Their growth rate's determined by Liebig's law of the minimum. They have no extra phenylalanine to excrete. On the other hand, if the microbe is limited by the amino acid, takes up all the amino acid and glucose, synthesizes amino acid proportional to its glucose intake, but then it's got extra, so it can excrete some of that amino acid. So to sum up here, if you're glucose limited, you don't excrete amino acid. If you're amino acid limited, then you do excrete amino acids. And this assumption has been verified. Tyler did some assays and it more or less matches. And so this is our model. Very similar to our previous work, except for the excretion term now depends on the difference between your glucose-limited growth rate and your actualized growth rate. And when we simulate the model, you might have noticed this earlier or not, these dashed lines are actually model outputs. They more or less match the experiments across the range of parameter values. But you might be thinking, on my list of mechanisms of population cycles, there was external forcing in this experiments in batch culture, which gets a big forcing every day. So we wanted to see if this was internally generated or just reflected the external forcing, this period 2 cycle. And so then we took our same model, same parameter values, and just ran it in continuous culture chemostat mode. And what we find is that, in fact, there is a robust area parameter space that gives rise to these internally generated limit cycles, so population cycles through mutualism. We want to delve a little bit more into the mechanism, how this works, and also to figure out under what conditions it occurs. So we took that 5 ODE system and simplified it just a little bit further with some more simplifying assumptions. And we were basically assume the glucose is constant, total abundance is constant, and we end up with a simpler system of 3 ODEs for the 2 amino acids, as well as F, which is the relative abundance of our 2 strains of bacteria. And based on parameter values, we found out that the resource dynamics are fast relative to the relative abundance dynamics. So we have this fast, slow dynamical system. So what we did then is we would study the fast resource subsystem as a function of the slow relative abundance variable. So in this case, We just have three possible equilibria. They all line up nicely. It's continuous and smooth. No cycles in this case. However, there's a case two, where we have this intermediate region where the two resource metabolic equilibria are stable and separated by a unstable equilibrium. And so this is the case where we can potentially get the limit cycles. And so what happens is it's basically a relaxation oscillator. So if you're on one metabolic state where one microbe is feeding the other, its relative abundance goes down over time, but we stay on this attractor until we reach this bifurcation point, in which case we do a quick jump onto the other attractor, and now they switch roles and move back the other way. This is what that looks like in time series form, but here's a little cartoon. So in this case, we're going to start off with this orange bacteria being amino acid limited, so it excretes amino acid for the other guy. The other guy, the blue one, is glucose limited and therefore has nothing left to excrete. So the red one is feeding the blue one. Blue one's population size increases until it can't, the orange one has shrunk so much it can't keep up with the demand of the blue. They switch roles and the cycle goes in reverse. Couple other things we looked at in this. What's the implication of this oscillations? Well, one thing we found is that it provides some sort of resistance to cheater strains. So this is a case where we have a stable equilibrium of the cross feeders. At time zero, we introduce a cheater. A little bit like in Dervish's talk, the cheater grows, drives the cross feeders down below their threshold, and then the whole system crashes. However, if we're in one of these oscillating systems, then when we introduce the cheaters at time zero, they decay away and they're kept out. And the reason for that is because there are only very small moments of time when both resources are available for the cheater. So the cheater's growth rate most of the time is negative. Finally, one of our reviewers, reviewer 2, I believe, had a great comment, great question. So they wanted to connect then this, continuous time limit cycle dynamics with the period 2 cycle that we found in our experiments. And so first thing you have to do to connect these two is you need to crack open the box. With the model, we don't just measure their abundance at the end of the cycle. We can look at, see what happens. So we see the dynamics within the serial batch culture. And then we just took our serial batch model and we changed the period of the dilution while keeping the total nutrient influx and dilution rate constant to make it a fair comparison. So this is a case where we start off close to the continuous system. And so here we have a nice 15.23 hour limit cycle. With 1 hour forcing, we get what appears to be just a fuzzed out version of that. Although technically it appears to be aperiodic. If we make the forcing a little bit slower, we have a 2 hour forcing period. Then we end up with what's technically a period 8 cycle. And you might notice 8 times 2 is 16, which is pretty close to the natural frequency of 15. So it kind of gets locked in there. As we make the forcing slower and slower, this limit cycle gets fuzzed out more and more. We go through a lot of complicated bifurcations until we get to about 12 hours and it starts to become qualitatively different when the abundance at the beginning of the cycle gets super low. So now we're getting into the more like the batch mode until we end up with the period 2 cycle that they saw in the lab. One thing that I think is really interesting is if they would have done their experiment with a 30-hour forcing for whatever reason, they would have just observed, what appears to be a stable equilibrium, and we never would have even gone down this whole path. And so this is summarized in this, very complicated bifurcation diagram. A lot of things happening on there, but in fact, you can connect the continuous and discrete time dynamics. So there are some conclusions, but I'll just say thanks to my collaborators.
I wonder how, if you've thought of playing with the costs of the amino acids and therefore the ratio of the costs, like some amino acids might be even more glucose limited than others.
Right.
And so.
Yeah, so with the cheater doesn't pay a cost of synthesizing, so that's why they're cheaters and take over. What turns out to be the important parameters that determine whether you get into the potential for cycles or not, it's the stoichiometric coefficients. So if the cross feeders are more efficient in producing than the requirements, there's some inequality with Qs. So basically that's there. So it's a little bit the cost.
That's beautiful results. I wonder how is it connected to the very old result which was obtained without cross-feeding, Jeff Huseman, Nature 1999, where they saw how with 3 essential nutrients you can get cycles, with 4 you can get chaos.
Sure.
And it looks like you can get chaos with 3, right? 1 primary and 2 essential amino acids, right? If I understood your chaos crossover picture correctly.
Yeah. So I'd say Jeff Hausman's model basically what fell into that category of intransitive cycles. So it's sort of the rock, paper, scissors.
Yeah.
So there's a little bit different category of cycles there. In terms of, the dimensionality of our system, we also have to get the chaos in that crazed bifurcation diagram. It also requires the external forcing.
Oh,
so that gives you some extra opportunity for chaos.
Okay, got it.
And
Actually, somebody else first.
I think you
So for the chaotic behavior, because I know the experimental stuff you were able to get 2-period stuff, but were you able to ever get like chaotic behavior in the experiment? And did you ever think of using that to like fine tune your model to like get like really, really close to the experiment, even though it was pretty close to experiment 2-period?
Yeah, I would say the experiments went first and the modeling came second. So maybe that could be a good idea for a follow-up. I'm not an experimentalist, certainly not this kind of experimentalist. So it's something I can recommend to Ophelia. That's a great idea.